Welcome, welcome. Good morning. Welcome to a webinar about connecting urban biodiversity. Come on in, really glad that you're here. We'll wait another minute or so as folks arrive, but just want to say welcome and really glad that you're here to talk with us about Connecting Urban Biodiversity Project and urban biodiversity generally. Come on in, get comfortable. Close tabs, turn your phone over, off, take a drink of whatever you're still drinking, whatever you're drinking here at 10 o'clock. And shall we get started, team? Let's do it. Well, welcome to the Connecting Urban Biodiversity Project webinar. Really glad that you're here. I'm Annie Burke, and I'm a proud member of the Together Bay Area team. And I'm really excited for this event and excited to see so many people here and so many people that are interested in this work that we're talking about today. So as we arrive and as I welcome you into this virtual space, pretending that we were in person, I want to share just a few things. One is, and you don't need me to say this, but there's a lot going on in the world, in our country, in our state, in each of our lives. And I'm not going to name all the things because we'd be here for hours. But I want you to know that we acknowledge that there's a lot going on and that being a human being right now is a full and rich experience. Second thing I want to say, just as we get situated here, is that people are nature. We're not apart from it, but a part of the natural world. We're not above or better than, we are nature. To some folks here, that's an obvious statement. That's something that you know in your bones. Yet it's not how the dominant Western culture has operated. And I think before we have this conversation, we need to name that in order to have that kind of conversation, unpack it, talk about it. Third thing is that we're choosing, and I'm going to speak on behalf of Together Bay Area right now, to meet this moment with in history and all the things that are going on with two ways of being, grounding and togethering. So let's remember the ground under our feet, the lands that sustain us, the air we breathe, the food we eat, the water we drink. Let's stay grounded in the urgent and patient work that we are all doing to take care of this place and the places that we live and work. And let's be together. Let's be in community. Let's reach out and welcome people into the conversation, into the work. Let's be together. So I invite you into the space today with those three things. There's a lot going on. People are nature, grounding and togethering. And speaking of togethering, I want to briefly introduce Together Bay Area. Some folks here probably know us well and others not yet. We're a regional coalition of organizations working for climate resilience and equity. And there's a lot I could say about this organization, like days and days of it, because it's a really awesome place to be and an awesome place to work. But for now, I just want to talk about two things. The first is that we facilitate a regional conservation strategy called the Conservation Lands Network. The CLN, Conservation Lands Network for short, is a regional strategy that sets goals, tracks progress, provides tools, and catalyzes on-the-ground implementation, on-the-ground land conservation in the San Francisco Bay Area. The CLN articulates two types of science-driven and community-driven, science-based, need more caffeine, science-based and community-driven goals, a set of 5 overarching regional goals, and a much larger set of habitat-specific goals. This is a 20-plus year project or initiative. The CLN started around 2005, which is, according to my math, 20 years ago. The first version 1.0, was released in 2011. There was a progress report in 2014. 2.0 version was released in 2019. And just last year, we released a progress report on the version 2.0. And we're going to be talking about that progress report at a webinar next Wednesday, same time. I'd say same channel, but it's probably a different Zoom link. And you're all invited. So I want to plug that. One of the things that I love about the CLN is that it's a dialogue that evolves over time. It's a dialogue about what's important and essential for biodiversity in the region. And it evolves as our thinking evolves, as the people sitting at the table evolves, as the world around us evolves. And so it's a really important piece of the CLN that I really love and that is relevant to what we're talking about today. So if I invite you to learn more about the CLN on our website, and there'll be a link in the chat in a second. Second thing about Together Bay Area is that we convene, coordinate, and catalyze conversations. And that's what we're doing with this project. And I want to I want to get to the project. So I'm just going to say, we're going to post the calendar of events that we have coming up at Together Bay Area in the chat. And we invite you to get involved and to join us at future events. Okay, now with all of that context, we're here to talk about the Connecting Urban Biodiversity Project. This has been a highly collaborative collaboration. Yes, I know that I'm using that word twice very intentionally, between Together Bay Area and the California Academy of Sciences. So I wanted to introduce you to the team because this team has been a lot of fun. So first, these three people. One of them is me, so I've already introduced myself, but also on the project team is Tom Robinson and Laura Rosenthal. And I want to also name that Michaela Nee on the Together Bay Area team is a part of this and working behind the scenes right now. And then with the Cal Academy, we have these fabulous people whose photos are amazing. And on this team, we have Dr. Rebecca Johnson, Allison Young, Olivia Van Dam, and Dr. Avery Hill. And you're going to learn and hear from each one of these folks in just a minute. So that's who's the project team is. Now we'd love to hear and we'd like to see who's here in the room with us today. And there's a lot of you, so I'm not going to go around the table. Please introduce yourself in the chat with your name, your organization, and your favorite animal who lives in cities. Who is your favorite urban dweller? And that there's no wrong answers. So really looking forward to hearing who's here, seeing who's here, and seeing what animals you're bringing into the space with us today. While you do that, I want to tell you about how this hour is going to go. First, we're going to hear from the project team, get an overview of the project from Rebecca Johnson, and then each member of the team is going to talk about some of the lessons that we learned through this project. There's a report that tells the whole story, and we'll drop a link in the chat to the whole report so that you can see from beginning to end. Today, we're really going to focus on lessons learned. And then after the presentations, we'll move into breakout rooms for each of the lessons learned, and team members will be in those rooms to ask questions or to answer questions and to have a conversation amongst you all. There'll be a chance for you to connect and learn from other attendees. And I just want to emphasize that we are all experts here and we are all learners. We all have something to learn and all something to teach. Speaking of experts and learners, now I'd like to welcome Dr. Rebecca Johnson to the stage. Rebecca wears multiple hats. They're literally invisible, but they are there. First as director of Center for Biodiversity and Community Science at the Cal Academy. She also serves on the Together Bay Area Board of Directors, which I feel very lucky to benefit from. And I officially declare her as a world-class iNaturalist observer. And so with that, I'll turn it over to Rebecca.
Thanks so much, Annie. Hi, everyone. It's so nice to be here with you all today and to really talk about this work that we've been working on for a number of years. It's really exciting to be able to share. So the Connecting Urban Biodiversity Project is about connecting the ways we have done and thought about conservation for decades to what we know now and what some have always known, about inclusive landscape conservation that includes all lands. So it's about connecting people, data, and land to build the most complete roadmap to conservation in the Bay Area. But it's really about valuing nature in the places where people live, and also valuing the experiences of people who live in cities. And by valuing, I mean counting, including, recognizing, um, all of those things. And when I talk about this project, like when I'm talking to anybody about this project over the past number of years, I always accidentally call it celebrating biodiversity instead of connecting biodiversity. And I was part of the team that came up with the name, but I always get it wrong. And I think because in the end, I think that's what we've tried to do with this work, to celebrate and meaningfully include and be in solidarity with urban nature and the organizations and people fighting to protect it, because we're all in this together. All right, next slide, Michaela. So this project really was born at the release of the CLN 2.0 in 2019. So this is a little Wayback Machine picture for all of you, when the team, the Together Bay Area and CLN team, was released CLN 2.0. So this was a celebration of incredible collaborative work, updating a regional planning tool and identifying what's protected and what's not, so we can strategize and work toward the goal of conserving 50% of lands in the region by 2050. This work was funded by the Moore Foundation and the California Coastal Conservancy. And Allison and I, along with Gio Rapacciolo, the former data scientist on our team, were sitting in the audience, and I was like, this is amazing. And next slide, Michaela, we saw this map, right? This is like a classic CLN map. And the thing that we noticed, or I noticed, and we all talked about, like whispered in the audience, was that this map is incredible. And all of those urban areas you can see are grayed out on this map. And the cultivated land and the urban land, like, don't really count in the CLN 2.0 as areas that have conservation potential. And so we thought, like, what if we could change that? And Allison and I have been working for years at that point to organize people to make and share biodiversity observations in those urban areas. So what if we could think together about ways to bring all of those data and connect those people to the CLN, and also bring those data into the CLN? So after the presentation, we pulled Annie and Tom aside, along with Moira McEnsby, formerly of the Coastal Conservancy, and we started hatching a plan about what we could do together. So next slide. So like I said, urban areas were not included in CLN 2.0, but priority streams that run through those areas were. You can see those on the map here. This is a little zoomed in version. With the little dark blue lines, those are priority streams. So with the support of Moira and Shalini Kanan at the California Coastal Conservancy, we proposed a project to work with community-based organizations and communities living and working in severely disadvantaged communities, as described by Prop 84. Those are the areas in red, along priority streams. So now you're looking at the red areas with a blue stream, to gather data for use in regional conservation planning in the CLN. And also those same data, like work with them to use those same data to answer local questions, and overall to investigate how to bring community collected data to the CLN. So next slide. So as we were doing that work, we identified folks we had already worked with and had great partnerships with, who were working in areas around those priority streams with communities living and working in those areas. So we worked with 3 organizations. And I have their visions and missions listed here. I encourage you to click through and learn more about these incredible organizations. So we worked in Richmond with Yes Nature to Neighborhoods, We collaborated with Vencel Alfred, Carlos Guerrero, Blanca Hernandez, and Luis Pacheco to do work in Richmond. We also worked with Salted Roots, and we collaborated with Madoka Hara, Jessica Knox, JR Darren Ariana Guerrero Nardone, Trisha Ong, and Risa Ophelia. And also Friends of Sassel Creek, where we worked with Kate Berlin and Elena Stanger. And these organizations were amazing to work with and were key to us really being able to work directly with communities who lived and care about the areas along these priority streams. So next slide. And what we did with these different organizations was, for example, with Yes, we worked in Richmond along Wildcat Creek in Alvarado Park to hold events. And this is a little bit of an outline. You're going to learn much more about this from the rest of the team, to make observations along Wildcat Creek with the Yes community. Next. And then we worked with Salted Roots and Friends of Sassel Creek in Oakland along Sassel Creek in Diamond Park to do the same, to learn about these places and make and share observations there. So next. So we engaged and partnered with and really learned from these awesome CBOs. We worked together to gather data in a couple different ways that you'll learn about next. Gather data about species in urban areas. And next slide. What we really did is strengthen relationships between people and land and nature, knowing that the relationships are really what this work is all about. And now we're excited to share with you more details about what I just outlined in the form of lessons. So I'm going to turn it over now to Olivia Van Dam, who is the community science coordinator with me at the California Academy of Sciences.
Hi, everyone. Thank you so much. I am excited to share lesson number 1 with you today through one of my favorite mediums, storytelling. So lesson 1 is that collaboration and local expertise are essential. This is essential because local residents and organizations hold valuable expertise about their environments, including knowledge of biodiversity, land ownership, and local politics. Building relationships takes time, and it's a very necessary investment for work in the Bay Area and beyond. CBOs are trusted connectors to their communities, and their involvement ensures that conservation efforts address local priorities, needs, and concerns. And this type of collaboration can also help address systemic environmental injustices and lack of access to local nature spaces. So I was really excited and honored to bring partnership and collaboration with these CBOs into this project. And so for my first story, I want to share about Yes, Nature to Neighborhoods. Next slide. So we worked with this community in really a special way. On the first night, which you can see on the very left, I'm going to just tell a story about the iNaturalist training. We use iNaturalist as a tool to help gather data and help folks make their observations of their local nature to collect the data for this project. So we went into, I went to the Yes Nature to Neighborhoods headquarters, which is in Richmond, California, on Friday evening. Allison tuned in on Zoom. I was there in person, and we did a demo of how to use iNaturalist, how to make your account, how to download the app. And that night during the training, one of the fathers and the families that was there, what was so beautiful about Yes programming is that it's intergenerational. And they have an amazing cohort of youth leaders called the C3 Youth Cohort. And those youth also bring their families, and they invited their families to come to the BioBlitz in Alvarado Park on Saturday. And so Friday night, we're there in the room with this family, And this dad, Juan Carrillo, he was so excited to learn about this tool because he said, I've been fishing along the bay shore and I've been taking photos of birds in my neighborhood in our garden, and I don't know what they are. And I've tried to use Google to find out what these animals are. And now I can just upload them to iNaturalist retroactively, and it can help me identify these fish and what I catch and what I see in my garden. And that really just stuck with me, and I wanted to share that because it leads to one of the main takeaways for me working with community is that everyone has an existing connection to their local place. Everyone has existing knowledge, and we were there to just offer a new tool to deepen that relationship and that existing knowledge. All right, the next story is with Salted Roots at the Friends of Sassel Creek in Diamond Park in Oakland. And this day was filled with joy and filled with a lot of amazing collaboration. And I was so excited about obviously being in nature with youth and their youth cohort, also the Rising Leaders cohort, and to see how folks were connecting with one another and with this creek and this watershed and getting their boots in the creek. And one of the fun parts about how we run our BioBlitzes is we actually try to do a data wrap up and share what everyone saw and do a share out and a lunch afterwards. And we did this with Yes as well. But during this data work breakout and sharing the live project on iNaturalist on the screen, as you can see here inside of the community center at Diamond Park, we were pulling up people's observations that they uploaded during lunch. And the screams of joy and the excitement to see, oh, that was my spider observation, or oh, that's amazing, who took that photo? And then giving props to Trisha for her amazing photography and observation, and seeing it live inside of the iNaturalist app. And then doing this beautiful poster where we wrote down folks' observations of the water striders and the salamanders and the butterflies, and just doing a popcorn share out in the room. The liveliness and the excitement and joy that Salted Roots really carries on through their mission in everything they do. And to see Kate's amazing expertise on Sassel Creek and how Friends of Sassel Creek can use this data and can use BioBlitzes for their future conservation of this watershed. It was just a really beautiful moment. And that led me to another key takeaway, that youth leadership is vital to community building. Youth are our current leaders. They are doing environmental work today and right now. They're not future leaders. I saw through their leadership in both of these organizations and how both of these organizations lead and want to develop the youth's skills and career pathways in this work. May we all continue to support youth in their growth and learn from them and grow alongside them. So those are takeaways from why collaboration and local expertise are essential. I'll pass it to Allison for lesson number 2.
Great. Thank you so much, Olivia. A quick note, if you're seeing my name on the screen right now. So for most of this project, I was co-directing the Center for Biodiversity and Community Science with Rebecca. I have recently moved into a job at iNaturalist, so still very tied to this project as well. But yeah, so I'm now with iNaturalist, which is why my name says that. So this our second lesson is that nature thrives in cities, and we really want to recognize the role of urban biodiversity. So like Rebecca was talking about when we went to that CLN 2.0 launch, we did notice that the urban areas were grayed out on those maps showing areas of conservation value. But when you look at the Bay Area and like in the satellite image, the urban parts of the Bay Area, the creeks that run through them, like the small green spaces in people's backyards, like that's what connects the Baylands to the uplands for most of the Bay Area. And it's really what we have to work with when we think about creating more connectivity from our shores to our ridgelines. Next slide. You know, and we know that there's nature in cities. Like, we're not denying that there's nature in cities. There's species that we've become really, really familiar with as our urban neighbors, like pigeons and little cute raccoon trash collectors, like this one here, that kind of blend into our everyday surroundings in cities. And then there's also species that we get really excited to see when they choose to make the places that we live are their homes too, like when peregrine falcons nest on tall office buildings in San Francisco or in the Campanile on the Berkeley campus. But generally, in traditional conservation work, urban areas are dismissed as places where native species, especially those whose primary habitat is outside of cities, can really flourish. Next slide. So we really wanted to change that narrative in this project, and not just by telling a different story. Like, we wanted to collect the data, which Rebecca will talk more about, and we wanted to run the species distribution models that Avery is going to talk about that would really show the wide variety of native species that are not only using urban habitats, but really thriving in them as well. And like Olivia just mentioned, we wanted to engage people and communities in all of this work, because we recognize that people who live in these places and they have knowledge to share. They've seen changes through time as they live as they've lived in these cities, and they also have information about the species that they know are their neighbors. And we know that there are opportunities to deepen people's connections to and understanding of nature in urban areas through this sort of work. So Rebecca and I had been holding BioBlitzes for years before we even started this project, teaching people how to look for and document species. And one thing that we always loved hearing when we held BioBlitzes, and when we hold BioBlitzes, and especially in urban areas, is people discovering a species in a place that they might like walk through or visit every day. Like, maybe they walk through Golden Gate Park on their way to work, you know, every morning, or they walk their dog in McLaren Park every afternoon. But through participating in a BioBlitz, where they're given this opportunity to really slow down and look for species and be really curious, like they find a slender salamander when they had no idea that salamanders were in that park or in San Francisco at all. We heard that all the time, like no idea that salamanders are in San Francisco. And then realizing as they continue to look under logs that these slender salamanders are actually everywhere and they're thriving in the city as well. So like one of our favorite things from BioBlitzes. Next slide. So through this project, we identified 19 Conservation Lands Network target species that seem to be doing really well in our cities, such as Nicklin shoulderband snail, which is a species that's endemic to California and actually considered critically imperiled. Alligator lizards, like this one here that's clearly gotten a job as an Uber driver in someone's car. Wavy leaf soap plants, western bluebirds, like this one, this little ranger, letting us know that there is no poop fairy, all doing really well in our cities. And then we also identified the characteristics of our urban areas that are helping these species thrive. So what are those things that are in our cities that are actually helping, that are creating habitat and helping these species survive in cities? So the findings from this project really, we're trying to challenge these outdated notions that cities are ecological deserts. And we really want to emphasize like the many ways and the many species, the many species and the many ways that species have adapted to urban environments. Cities really are vital connectors, bridging our natural areas like our baylands to our uplands and supporting like a really diverse suite of species. And we recognizing that urban spaces have value for biodiversity is really tied to creating more inclusive conservation strategies. You know, moving forward, incorporating urban areas into conservation planning is something that we don't see just as an opportunity, but really as a necessity going forward. So with that, I'm going to turn it over to Rebecca to talk about how we collected the data and how we involved communities in that process.
All right, thanks, Allison. So I'm just going to really briefly talk about the data that Allison mentioned. So this map is like, it looks like one of those things that you have to focus your eyes to try to find the secret picture hidden behind. But this is a zoomed in picture of all of the iNaturalist observations that have been made in the San Francisco Bay Area. Each one of those little markers is an observation. And so it's amazing the amount of data that people volunteering their time, people who love nature, have made to just take a picture and upload that to iNaturalist. So there's a picture of a guy here making a little iNaturalist observation. I want you to think about every one of those dots represents someone doing this, right? And having that connection. And that's incredible that this like the largest source of biodiversity data, point biodiversity data that we have for the Bay Area and actually for the world is through an activity, like someone just taking a picture. So Allison, like Allison said, through the work of our team here at the Academy for almost over a decade, worked to provide opportunities, to provide opportunities for people to make and share observations like this. We can't take credit for like every single one of these observations, but we worked for and still continue to work to encourage people to make these observations. So next slide. So when we were looking at the CLN, and when you think about the CLN, here's just a zoomed out, like these blue area is like a conservation prior area, like where we think about conservation in the current CLN. And then these areas that are gray are not. And next slide. If you look at those data again, you can see that like all the bulk of the iNaturalist observations are made in the areas that were not considered in the CLN. So this is just a like distillation of the realization that Allison and I had about how these data could be and should be used, not only to understand where species are in conservation lands currently, but also in urban areas. So we use two tactics to collect more data around species in urban areas. So next slide. So Allison laid out for you the 19 different species that we were really focusing on, and Avery will talk about how we really used data on these species. But in order to collect more data about those 19 species and groups, we launched a campaign in 2023 called the Bay Area Urban Species Search. And what we did is ask all of you and and people everywhere to look for and make observations of those 19 species and species groups over this window of time. And so some folks held events, and so we want to thank those folks. Yes, Nature to Neighborhoods, Keep Coyote Creek Beautiful, the Trust for Public Land, and San Jose City College. But a lot of people just took this as like a little like a treasure hunt. Let me go look for these species where I live. And over 15 days, almost 1,000 people made 2,461 observations of 58 species. So although we have 19 species groups, some of them include many species. So this was a really nice way for us to gather new data in places about certain species in places where people live, and also just to get the word out about our campaign. Next slide. And then as Olivia detailed, we held two events, one in Alvarado Park in Richmond with Yes, and the other at Saucil Creek with Salted Roots and Friends of Saucil Creek. And so we collected observations in those specific areas along those priority streams, looking for every species, but especially looking for those target species. And as we analyze and look at all these data, I'm going to turn it over to Avery to talk about this next. We use those new data that we collected and also all of the data that already existed on iNaturalist. All right, so I'm going to turn it over to Avery to talk about how we modeled species distributions of these species for use in the Conservation Lands Network.
Great. Thanks, Rebecca. Hi, everybody. Avery Hill. I was responsible for turning a lot of these community collected species observations into range maps and other kind of conservation relevant biodiversity insights across the Bay Area. If the word species distribution modeling sounds scary, I think it's important to, my perspective is that all range maps you've ever seen, any species kind of like map you've ever seen, is a species distribution model. And historically, this used to be done by an expert going out and looking at observations of where they find this plant or this insect, and then going back and thinking, okay, well, where's the other kind of habitats that are similar to where we found this species today? Now, today, we use more kind of statistical, computational techniques, but the kind of foundation of thinking about where species are, where you find them, and where you could potentially find them is kind of the same. And because we can't know where all species are at any given time, even if we have millions of iNaturalist users taking observations all the time, we're never going to know where all the species individuals are at any given time. Next slide. So what we did is we used environmental data from across the Bay Area, 18 different kind of variables, climate variables, traffic, pollution, tree cover, things like that. And then we took each species observation that we collected through these campaigns and our various communities collected and associated these different environmental kind of variables with each of these observations, created a model that kind of related all of these observations to the environment, and then used that to kind of estimate, where we expect to find these species, what the kind of suitable habitat is for these species across the Bay Area. Next slide. And so there are two, I think, main advantages of using the species distribution modeling technique for urban biodiversity efforts. One is pretty obvious, and I'll demonstrate it here with the shoulder band snail, my favorite urban dwelling species that you can find in urban areas. Um, uh, with species distribution models, we can get range estimations, range maps, and I don't really need to talk about the usefulness of understanding where a species is and where it isn't, and or where a species could be, and where the habitats are that it doesn't really like. I think that should be pretty clear. How that's useful to know those things about species that you care about. Another useful, and on the left here in this map, the white dots are where we actually found this species, the shoulderman snail. And in that green that you can kind of see, that's where the habitat's really similar to where we found them in those white dots. And where, even if we didn't see the shoulderman snail there, we expect them to be either in that area or just suitable for that habitat. If you wouldn't mind clicking the slide. Another exciting kind of component of this is not only can we develop a good sense of where these species are, we can also get some insight into what different environmental or habitat features are responsible for, the shoulder band snail being in particular places. So for our shoulder band snail, far and away, the distance to 10-acre green space, so 10-acre size or larger green space, was a really important determinant of whether we see shoulder band snails. It's much more important than something like elevation or distance to kind of standing freshwater. And we can zoom in even further, if you mind clicking one more time. And we can look at kind of the relationship between habitats, the 10-acre green space, and habitat suitability, where this snail really liked to be in actually 0 meters away from 10-acre green space. So it likes being in 10-acre green spaces. And we can, with this modeling technique, also understand that if you're greater than 2 kilometers away from 10-acre green space, you're unlikely to see this snail. So these are important insights for protecting, this particular species. If you don't mind clicking one more time. We aggregated results across these species distribution models across our species, and you can get kind of regional insights into what different environmental variables are important kind of regionally. This is very dependent on the set of species that we have, but looking at kind of the regional environmental variables that we have some control over, 10-acre green space is up there, as well as land cover class and kind of the percent tree cover. Next slide. And so as Rebecca kind of talked about a bit, our set of species that we did this modeling from, something that we learned is that, we were opportunistic in kind of what species we had available data for, but we can be really intentional, I think, in future iterations of this project, or we can really leverage species distribution models for specific species that have specific kind of conservation goals and sets of species that we think indicate different important aspects of our regional urban biodiversity. Next slide. And finally, these species distribution models are really useful in isolation, but they're even more useful when we consider them alongside other important conservation decision-making data and tools. The Conservation Lands Network is a perfect kind of home for a bunch of conservation relevant decision-making data, and CLN 3.0, the plan is to incorporate species distribution models into this so that you can for the benefit of the species distribution models and for the benefit of the CLN. Great, that's it.
Excellent. Thank you, Avery. Hi, everyone. I'm Tom Robinson. I was fortunate enough to serve as project manager for this incredibly rewarding exploration with the folks you're hearing from today. I'm excited to share about lesson 5. Cities are complex landscapes, and that we should be thinking about engaging municipalities in particular deliberately. So as you've heard, a key theme today is that urban areas are not ecological deserts. Native species actually make them a part of the regional ecosystem. But cities are more complex in almost all ways than unincorporated kind of rural areas. Where a lot of our existing priorities exist, many more jurisdictions, stakeholders, et cetera. And this complexity represents some unique challenges for conservation. Therefore, urban biodiversity work requires deliberate engagement with municipalities to understand and work with that complexity. And I want to share just 3 finer points to this particular lesson we learned. The first is demands on resources. Cities manage multiple priorities: housing, transportation, infrastructure, public safety, typically making biodiversity a lower priority. Municipal budgets and staff capacity often limit the focus on ecological stewardship. For example, park and recreation departments may only be able to focus on maintenance rather than habitat restoration. If we want to integrate urban nature into conservation strategies, we have to tailor our approach to each city's structure and priorities. Number 2 is a very unique relationship to the land base in cities. Unlike rural areas, urban lands are fragmented. And then also have, again, these overlapping jurisdictions. Private landowners, public agencies, and community groups all play roles in land stewardship in urban areas. For example, a successful creek restoration may require coordination between public works departments, environmental services, parks and rec, and then local community-based organizations. So there's a unique relationship. Number 3 is that cities aren't universal. The context varies from city to city. Each municipality is structured differently. Conservation leadership may sit in various departments. So in one city, you might have public health leading greening efforts for climate resilience. In another city, public works manages urban trees and stormwater. And in a third city, sustainability offices might drive biodiversity planning. We had some great conversations with folks at East Bay Regional Park District. This is a park agency that manages several urban parks. And these conversations showed that engaging the right department is key, but that differs across the various cities. Next slide. So the take home for us is really kind of, thinking of all of that in mind. Our hats are off to managers of existing urban biodiversity programs. I think of the City of Oakland's Watershed and Stormwater Management Division, Sonoma County Ag and Open Space District's city-based matching grant program. Santa Clara Valley Open Space Authority has a similar matching grant program where they work with cities. These are programs that really back with local funding and action that the idea that urban green spaces are not just amenities, and in fact, they are critical links in a larger biodiversity network that connects the bay to surrounding uplands. So moving forward, we want to ask this question: how can regional conservation planning support making biodiversity a mainstream priority? And we believe at least, and at least up to part of that answer is understanding local government structures and aligning biodiversity goals with city priorities. And in order to engage, it's crucial that all other interested groups understand those priorities, which again are unique to the city in question and its social and financial context. And a key ingredient to the success of urban biodiversity is early and deliberate engagement with municipalities in order to foster long-term partnerships and ultimately long-term impact. So now I'm going to pass back to all the way back to Annie to move us into the interactive part of our hour. Annie.
Awesome. Thank you, Tom, Avery, Rebecca, Allison, Olivia. Really loved hearing from each of you about lessons learned and stories and data and pictures. So thank you. As I said at the start, one of my favorite things about the Conservation of the Lands Network is that it's a dialogue. It's a conversation we're constantly having, constantly evolving. And there are people who have been involved in the conversation for 20 years, and there's others who are just joining, and we welcome all of those perspectives. So here's what we're going to do next. There's a lot of you here. Otherwise, we would just ask you to raise your hand and we'd have a conversation, but that we can't do very well. So we're going to do two things. We're going to invite you to join a breakout room. Each of the presenters today will go to one of the breakout rooms. The breakout rooms are assigned by the lesson that they presented on. And you can move yourself to one of those breakout rooms and engage in a conversation there and ask questions. We also have set up a Canva whiteboard. Imagine, that we have access to one big virtual whiteboard. And on that, you can see if you click on the link in the chat, you can see different sections, different colored Post-it notes. If you click on one of those Post-it notes, you can write a question or share a link or share an observation. And the different colors are for the different lessons that we went over. So there's collaboration and local expertise, the role of urban biodiversity, community science as a powerful tool, species distribution modeling, and engaging municipalities. And we invite you to go in there and ask your questions or share information. And also moving into breakout rooms. So with that, I invite you to click on the link at the bottom of your Zoom screen that says breakout rooms, and you can move yourself to the breakout room of your choice. Hi, everybody. Welcome back.
We were mid-sentence,
unfortunately. Mid-sentence. I'm going to say to prioritize conservation, like for my team, like what does it mean to really do that in an equitable way, right? Like, I think those are really great questions. So sorry, everyone has to hear the end of.
No, as conveners, we love it when nobody wants to come back to the main room because it means that you're having really interesting, valuable conversations in the small room. So thank you for contributing, participating, for having full, rich conversations in your breakout rooms. I want to encourage you to add your notes, your questions, your reflections into the Canva, to the whiteboard that we shared earlier and that we'll share again. This is information that we will use going forward. We're going to incorporate your input and feedback and reactions from today's webinar into future conversations about urban biodiversity and regional conservation strategies. So it's helpful for those of us who are here today, but it's also will be used going forward. I want to wrap up our session today and with some invitations and some gratitude. First, we invite you to read the full report. You got a taste of what we did and a taste of how we did it today. And we encourage you to read the full report, which you can find at togetherbayarea.org/habitat-goals. We also invite you to take pictures of nature and cities, download and use iNaturalist, although I have a suspicion that most people here already do. I'm going to add another invitation, which is somebody needs to create the shoulder band snail emoji. So I'm just going to put that out there. There's somebody here that can do that. So that's another invitation. Join the Cal Academy event or campaign. You can learn more about the community science program there. Work with community-based organizations. There's so much that they can share and contribute. And think about cities when you're thinking about regional biodiversity. Coming up to a Zoom room near you soon is the CLN 3.0 project, which this Connecting Urban Biodiversity or Celebrating Urban Biodiversity project will help inform. So you'll hear from us and Together Bay Area with more information about that project coming up soon. And with that, I want to say thank you to everyone who came and participated and who is writing notes on Canva whiteboard right now, and for all the work that you're doing to connect and celebrate urban biodiversity in the Bay Area. Really appreciate the work that you're doing. Appreciate you being here today. And on behalf of everybody on the project team, thank you for coming, and we look forward to being in conversation with you going forward. And that's a wrap.
Thanks, everyone.
Thank you.
Thank you.